Live from the North Carolina Museum of Natural Sciences in beautiful downtown Raleigh, it's the Science Cafe with your host, Chris Smith.
Good evening, everybody. Welcome to the North Carolina Museum of Natural Sciences. Hi, how's everybody doing today? Raise one hand if you're doing great. Raise the other hand if you love science. And then go, woo! Fantastic! It sounds like everybody's doing great this Thursday evening. Welcome to the museum. Welcome to our Science Cafe. This is our weekly event, actually, where we give you, our Triangle residents, our science community, the chance to meet some scientists. In this case, tonight, actually meet some scientists and some science students, learn a little bit about what's going on in the world of science in and around the Triangle, and maybe find out how it applies to your daily life. That's what the Science Cafe is all about. I really love the Science Cafe format, right? So, number one, you can get dinner here in the Daily Planet Cafe. You can get a beer, as several people are like, don't forget the beer. Yeah, you can get a drink, and the cafe is great. How many of you are friends of the museum? Museum members? Wonderful. I see lots of hands up. Thank you so much for that. If you're a friend of the museum, if you're a museum member, you actually get discounts here in the Daily Planet Cafe, and then a whole bunch of other museum stuff as well. So you can get brochures, talk to me about that later if you like. I can definitely fill you in on how you can give money to the museum and help us out. Yeah, and those funds, of course, support things like the Science Cafe, lots of events that go on here at the museum, and we really do appreciate the support. And it goes a long way to helping us share what's going on in the science community with you, right? We've got this great, big, giant museum right here in the middle of Raleigh, and now we're something like the second largest natural history museum in all of the southeastern U.S. We just got beat, I learned this, we just got beat by Miami, who opened a new museum on Monday. That's just a little bit bigger than ours. But anyway, whatever. Tonight, I'm proud to bring you actually one of our museum scientists, Adrian Smith. He's head of the Evolutionary Biology Lab here at the museum, and his topic of research, the thing that he loves maybe more than his one-year-old son, no, okay, are ants. That's why I wore my special ant shirt tonight. And so give Adrian a round of applause. Welcome him to the stage for the Science Cafe.
Thanks, Chris. Thanks for coming out. This is going to be an interesting presentation. It's going to be something that you normally wouldn't see at a Science Cafe, but I think you're going to like it. It's all about bio-inspired design. So let me sort of define what I mean by that. Has anybody heard of the field of biomimicry? A few people. Okay, great. So biomimicry and bio-inspired design, basically the same sort of thing. That's what this is going to be about. So it's about approaching science from the standpoint of design and the arts. And so how do those things intermix, and how can we get sort of design ideas, human innovation ideas, from looking at nature as a natural model? So one definition that people use for what biomimicry actually is this idea that nature, looking at how organisms solve problems, nature can be used as a model, a mentor, and a measure for sustainable human design. So that's sort of the general framework that we're going to be working in tonight. And it's not going to be me presenting, so it's going to be a group of students doing lightning talks, about 5-minute talks. They're talking about their project that they did in a class that was actually taught at NC State and also here at the museum. So, is biomimicry a new idea? No, it's not. We've been doing it for a long time, right? If you think back to ancient Greek mythology, when you think about one of the oldest examples is humans striving to fly, right? And if you go back in the literature and in mythology, flying is connected to looking at organisms. How do they do it, right? How do birds fly? So see depictions of flying using bird-like wings on humans, right? If you go up to the Renaissance Da Vinci, look in his codex on flight, you see a bunch of sketches, a bunch of ideas about how flight works in nature, how birds fly. And then if you go into the 19th century, some of the earliest innovations in actual human flight are from studying gliding birds. Here you see a glider based on how birds soar, right? So it's old examples of biomimicry, bio-inspired design, to try to see how nature does things that we don't do, and maybe does things better than what we currently do. Here are actually two of the most famous examples of biomimicry. And I'm gonna quiz you on this. So these are two things that are present in this room, technology that's in this room, that is based on looking for nature as a model. So the one on the left here, does anybody guess what that is? The wasp. What is that an organism a model for? Nope. I mean, yes, but that's not what I'm looking for, but close. So what was that? Nope, close. So it's actually connected to the honeycomb. It's paper. So paper from wood pulp. The idea from that was actually sort of originated from looking at paper wasps, who actually will bite off pieces of wood and mulch it together and then make their carton nests out of paper. So before that, we actually used paper that was made from things that weren't wood-based, right? So the idea of using wood-based mulch for paper came from wasps. How about the second one there? These are burdock seeds. Exactly, Velcro. Velcro is one of the most famous examples of biomimicry. A guy was walking his dog, got these seeds stuck in his dog's hair, and he's like, oh, this is a really interesting attachment mechanism. And there you get Velcro born out of that. So 2 examples of biomimicry every day in your life, right there. So usually when people go out to do biomimicry, it's from the design side. It's designers sort of asking, oh, how does nature solve a problem? How can I use that in my product innovation, in my personal work? And usually to do that, they go and they sort of look at the biological literature, look at examples of how nature solves problems, and use that as a basis for putting into their design. But that really has a major assumption in it. It's that the major assumption there is that nature is something we can look for ideas about how things work, because we know how nature works. And really the answer to that is most of nature is unknown and undescribed, right? Mostly we don't know how things perform particular tasks in nature. A lot of nature is undescribed and unstudied in any sort of meaningful way. So we took this as an approach, an experimental approach, for teaching our course in biomimicry. We asked these 2 main questions. These are 2 main questions that guided everything that you'll be seeing later on tonight. What would happen if designers and biologists combined their research processes to explore functional biodiversity? So we know that there's a lot of species out there, a lot of diversity, and all those species are well adapted for their environment. So how can we learn from that wealth of information, untapped information out there? The second point here is, what if innovative design thinking guided the process of scientific inquiry and design solutions were generated alongside scientific discovery? This is the main point that guided our course and guided these projects that you'll be seeing right now. We took designers, put them in the lab here, put them in the lab actually on the 3rd floor up here in my lab, and asked them to bring their design expertise, their design lenses, to look at how nature solves problems. So the course that you'll be seeing here is an experimental course that we just taught for the first time. It's the first time any course like this has been offered at NC State. And it was taught with 2 other instructors, Dr. Clint Pennick, who's right here. He's a scientist here at the museum and also at NC State. And then an architect, Dr. Sarah Glee Queen, who can't be here tonight. But she was our architect design instructor. And the students pull from a wide range of expertise. Most of them are in the design and arts. A few of them are environmental scientists. But most of the students you'll be hearing talking, this was their first sort of scientific exploration about an idea. And it was really neat to see them go through that process and then derive sort of innovative ideas. And really, even in the process of doing that, push the scientific boundaries forward. So all the talks that you'll be seeing tonight are based on ants. You might ask, why ants? I don't love them more than my 1-year-old, but they're a close second. Probably 3rd behind my wife. Let's scratch the record on that one. So, believe it or not, there are 16,000 described species of ants. 16,000. That means 16,000 ants that have a name, have a genus and a species name. There's not that many ant scientists in the world. A lot of these ants have never been looked at, never been studied. So ants are a huge sort of boon for looking at diverse solutions to common problems. And we looked at them for that because they share a lot of common problems with us, because they're all social animals. They live in a society. So they live in these dense groups of related individuals, and all of them work together to form a functional colony. They divide labor. They go out and get food to cooperatively raise their young. So what we did to sort of jumpstart these student projects, about 1/3 of the class that we taught was devoted solely to the development of these projects. The rest of it was an introduction to the field. so to jumpstart those projects, we gave them areas of ant biodiversity that are really interesting and sort of undescribed and undiscovered, and really not that much research existed. So we gave them a problem. We said, here's this really cool thing that we know ants do. They build homes to live in. They're masters at collective behavior. They cooperate to do things like raise their young, get food. They are really good at caring for brood and cooperatively raising their young in their nests. And also, they look crazy, so they have crazy patterns on their cuticle. We gave them these 4 areas and said, here, we want you to study this as the designer working in the scientific realm. So without further ado, I'm going to introduce some of the student groups, and they're going to tell you 5 minutes, 5 or 6 or 7 minutes about their projects here. And then we can go and answer questions at the end. First one, ant architecture.
Hi, I'm Kevin de Montbrun. I am a senior in architecture.
And my name is Hannah Frank. I'm a senior in sustainable agriculture and political science. So our project is called Ant-Architecture. So as kind of a starting point, we were really interested this semester in comparing how ants build their nests to how humans build their cities and their living spaces. And we kind of thought there was like no way we could find that there's that much that these 2 systems have in common, but that was kind of our jumping off point to see what sort of comparisons we could find and how those comparisons could maybe show some potential for like inspiration in design challenges. And so as Adrian was saying, ant nest architecture is still a very new field of research, and so it certainly has never been compared to human architecture before, which made our project a little bit challenging but very interesting. A lot in my hand right now. So just for a reference, this is pretty much what all the existing ant nest architecture looks like right now. All of their nests are made up of a series of tunnels and chambers, and all of this data pretty much comes from Florida State University. Walter Schinkel has pretty much done all this, and you can see, we'll talk about it later, but our class got to do a little bit of it this semester. So we kind of started with the assumption that
Hi, I'm Elizabeth Macias. I'm a student in architecture. This is a group project, but my partner could not make it. He's in technology, engineering, and design. And we will be talking about stigmergy in ant architecture. So to compare the architecture of ants and humans, we can say that we both build for shelter, and we create our spaces using the elements of circulation, such as corridors or shafts, as well as spaces for destination or storage, such as rooms and chambers. One difference is that ants do not follow a blueprint. So how is it that they build these very intricate structures? How do they self-organize themselves? So it is through stigmergy, which is this concept, is the idea of this indirect coordination between individuals as they interact with the environment. This term was coined by a French biologist as he was studying termites. As you can see here, this is a termite mound, and it was created by stigmergy. And essentially, it is the idea that an ant or a termite will build or will pile where an ant has already piled sand, or they will dig where an ant or termite has already dug. The literature about stigmergy, I guess, has been shown by Walter Schinkel through this layering of sand, where it shows this segmental transportation of sand pallets, which basically you can see the little pallets of sand containing multiple colors, and that's stating that one pallet has been moved throughout the nest and carried by multiple ants and not just one. So there's this indirect coordination happening between the ants. We decided, because in this area experiments are limited, we decided to conduct our own experiments. We used this typical setup of compacting sand in petri dishes, and we used the harvester ant, or the Pogonomyrmex badius, which is found in Florida as well as here in North Carolina. And here we were just observing ants digging. So with our, I guess, the second experiment, we will be able to, I guess, point out certain points, and those are that there's this randomization of tunnels as ants are digging, and also there's this fluctuation of spaces that the ants are building. And you can see in the video how a wall will be filled and refilled through this indirect coordination between the ants. And we saw that multiple times in running this experiment multiple times. So could there be an external force or body that can influence where an ant will dig? Or in other words, can somebody else that's not an ant stigmatize the ants? We conducted another experiment where we created 2 initial tunnels. So in a way, we were trying to stigmatize the ants. And what we found was the following, that the ants actually begin to dig where we started the tunnels. So I guess we saw that ants create their spaces through this self-organization process, and with this external force that was actually stigmatizing them. Could we apply this concept of shifting the direction of ants on people? So for example, we look at this empty space on campus. This is actually a new space that was designed because they tore down a circular building on campus. And right now, this space is not used at all. And with our experiment, we will try to make people use this space, in a way stigmatize this space. Here you can see that the spaces are not really used much, except for a little bit of circulation. And here, this video shows we actually used ourselves as the actors, where we would become that interaction in the environment to tell people to use the space. So we use our bodies as this trace left in the environment. And we were the people that were sitting on the lawn initially, and we saw that towards the end, people were actually sitting in the lawn. And we have another video of that where we increase the number of people. And we found that increasing the number of people also increased the number of users of this space that was once not used at all. Here's a table that shows sort of the correlation between when we the actors were using the space and other people were following what we were doing. So how can this concept be used in the designing of a space? And we actually realize that this is a concept that is being used currently. And in urban design, they use a term such as guerrilla urbaning, or activism, hacktivism. And these are examples where this minimal trace in the environment can change an empty lot into a lot that has been used. For example, this Wandering Box by Matt Tomasulo, where they brought this box that would sell beer and transform this empty lot by camp in Raleigh, North Carolina. And then events such as these by the Urban Raleigh Rangers, where they decided to hold events where people could hang their hammocks, and they made this plaza that was fairly unused more used through this event. So these are attempts at indirectly using a space to change the perception of people to use a space more. And another one where they show flags of people using the flags to show where they have gone on Dorothea Dix Park. Another one by Matt Tomasulo. So this sort of raises the question as how can you use this concept learned in ants, like stigmergy, to create and design public spaces in a local level that fluctuates in the way that the ants do as they self-organize their spaces. That's all.
Hi everyone, my name is Dipali. I'm a senior in industrial design, and this is my partner Lisa, a senior in graphic design, and we're here to talk to you today about the tactility of ants. So if you're like most people, you've probably underestimated the complexity of ants because they all pretty much look the same on the ground. But little did you know, there are over 16,000 known species of ants, and when you zoom in, you can actually see that they're very different. And so after observing these, we got really interested and wanted to take a look closer. So we looked at them through Z-stacked microscope images, as well as antweb.org archives to see the photographic documentation of these ant head cuticle structures, which, if you look at the screens, are actually pretty cool. So after these observations, we decided to ask ourselves this overarching question of what are the functions of ant cuticle structures?
Time to put our scientific caps on. Using scale as our foundation of study, we went down two main avenues of experimental research. So first, we compared ant cuticles to other known creatures with functional cuticle structures, so such as the shark skin, which is known to inhibit bacterial growth, the scorpion skin, which is erosion resistant, and the cicada wing, which kills bacteria. And then secondly, we also wanted to compare ant cuticle structures to other ant cuticle structures, so within the same genus as well as outside of the genus. So to focus on just the ant head patterns, we translated the 3D cuticle structures to a 2D graphic representation. So on the screen, you can see one of our ant head patterns. It is the Polyrhacus decora. We use this specific one as a point of comparison to compare to other cuticle structures. Mainly because out of our sample size of 19 that we looked at, this was probably the most average sized. So based on the function of our three precedents, so again, the shark, the scorpion, and cicada, we formed three hypotheses to see if the ant cuticle structures function in a similar manner.
So our first hypothesis was based on the shark skin, as you can see on the screen, which has been studied for bacterial inhibition. So the way it works is that the shark skin at a microscopic level has these diamond-shaped cuticle patterns with ridges, and all the scales overlap. So this makes it so that there's a small enough surface area for where bacteria can maybe reside, but does not propagate on the ants. So it made us wonder, can ant cuticles inhibit the growth of bacteria?
So actually not really, because when we actually did scale down our ant head reference to the shark skin to be relative to one another, we can clearly see that the shark skin pattern is a lot smaller than the ant head cuticle pattern. So it's approximately about 2 shark skin units to fit into a single groove of the ant head pattern. So from that, we can conclude that they do not share the same function.
Our second hypothesis dealt with the desert scorpion, and the way their cuticle structure works is that it resists erosion based on the bumps, grooves, and the protrusions from its skin. So we asked ourselves again, are ant cuticles erosion resistant?
And again, no. So the scorpion skin, once we scale it down, it's even smaller than the shark skin. So the ant cuticle head pattern you see next to it, it takes about 10 units of the scorpion skin to fit into a single groove. So again, we can conclude that they do not share the same function.
And so our third hypothesis dealt with the cicada wing. And the way the cicada wing works is it does not only inhibit the growth of bacteria, but it actually punctures bacteria and kills them completely. So we asked ourselves, can ant cuticles kill bacteria?
And as you can probably guess by now, the answer is no, they do not. The cicada wing is actually the smallest out of the 3 that we looked at. So they're just a bunch of tiny dots. I don't know if you can see it. It probably takes well over 100 of those individual pillars to make up a single groove on the ant cuticle head. So again, from that, we can conclude that they do not share the same function.
So after disproving our first 3 hypotheses, we decided to take our second avenue of research and go back to our ants and say, do ant cuticle structures have similar functions to those within the same genus? So we took our sample size of 19 ants and the 2D graphic representations of them and divided them between the genus Polyrhacus and then samples that were not from the genus Polyrhacus to see if we could find any point of comparison. And while we were not able to observe any underlying pattern within a single genus, we did observe underlying patterns across genera as well as species that were very distinct in their physical characteristics.
So when we started grouping them by physical characteristics, we determined 3 main pattern families across the different genuses or genera and species. So we isolated 3 main ones. So the network, which has a lot of connection points, linear, which has just a bunch of lines parallel to one another, and cluster, which is just a bunch of polygon shapes in a nested pattern. Because these patterns are not genera or species specific, so they range across different genera and species, we can infer that these patterns arose independently for evolutionary purposes. So basically, we can say that ant cuticle patterns are so distinct, they must have a function. And while we yet do not know what exactly that function is, we know that they do function in some way. So for example, they do convey some sort of information to those who study them. So if we were to use ant cuticle structures as an apparatus of texture-based communication, we can perhaps find applications to apply to a human system. So for example, if we were to, again, use it as a tactile-based communication system, we are trying to think about what kind of users would benefit the most from a tactile-based communication. So for example, the visually impaired. So existing design systems right now that use texture as a way to communicate information is stuff like Braille or rumble strips on the side of highways that tell you when you're going off the road, or tactile paving systems, which tell pedestrians about like impending danger or just to watch out.
And so with this idea of texture-based application in mind, we wanted to start prototyping with physical mediums like 3D printing or linoleum prints, just to see if there was any way that we could test this at a human scale and test things like texture or depth within the z-axis and see how those things affected the interplay between the grooves of the ant cuticle structures. And so on the screen here is an example of what might be a refined tactile paving system in which we extrapolate the cuticles from the ant cuticles from the ants, and we abstract them into these wayfinding tiles for visually impaired users to soon be implemented into a larger human system. And that's it.
Thank you.
Hello. My name is Allie Kenlin. I am a graduating senior, and I study a mixture of environmental science, communication, and policy.
And I'm Connor Lenhart, and I'm a rising junior in graphic design, and today we're going to be talking about bacteria and ants. And so the question that really got our whole sort of project started, we were observing these ants in the lab, and we saw these cocoons that sort of contain the ants' babies, and we saw this sort of black dot at the end of it, and we had no idea what that black dot was.
So we did a little digging, and what we found was that black dot is actually present in the larva of the ant. You can see that in the picture on the left. And that's actually called the meconium. So if you're familiar with that word in humans, the meconium is actually the first feces of the ant or person. And it contains all of that digestive material that's inside of the ant in its larval stage. And so after a little while, the ant goes into, the larva starts to spin a cocoon around itself. It goes into its pupal form, so that is the pupal case around it. And then within the first day of spinning that cocoon, the meconium emerges at the end. So essentially, the pupa lives in this cocoon with its own feces for up to a month inside the colony with all of the other ants moving about it. So we thought that was kind of strange.
Yeah, since most of the ants come from the same queen, they share a similar genetic structure, which makes them very vulnerable to disease. Like one disease could potentially like wipe out the whole nest, which happens. So, you know, having an ant live in this pupal case with like its feces at the end of it for a whole month next to all these other ants in this like close quarters of the nest could potentially be an issue. So we were interested in how they sort of stay sanitary and keep disease from spreading, having this feces at the end of the pupal case.
So we did some background research, and in our research we couldn't find a lot of information about the pupal case or any of its properties or the meconium itself. But one of our studies did find that in ant colonies, when a fungal infection would sweep through the nest, the ants would take out any of the larvae that hadn't spun their cocoon yet, and they would remove them from the nest because they didn't want any chances of them getting infected and infecting the rest of the colony. But they left anything that was still in its cocoon. So that kind of suggests that there's some properties of the cocoon that keep the pupa from getting infected and keeping them from becoming disease vectors within the colony. And then another study that we found, so we didn't find a lot of information about the ants, so we looked to, for inspiration from other insects. And so we found that in the bee wolf digger wasp, they actually have a symbiotic relationship with bacteria that fight off other disease-causing bacteria. So they inoculate their larval cocoons with this bacteria, and it provides a good relationship.
So to sort of summarize our background research, we put it in like one question that we were interested in doing more research on, which is, are there potentially any microbes or bacteria present on the pupal cocoon that sort of help the pupa avoid being disease vectors within the colony, especially since they're living in these close quarters with the feces again at the end of the cocoon?
So for our experiments, we were fortunate enough, we got to borrow some of the pupal cases from some of Adrian's colonies in his lab. And the way we sampled them was we literally split them in half. So the top half doesn't have the meconium, and the bottom half does have the meconium. So we took samples of anything that would be living on the outside of the pupal case, and we plated them so that that would allow any bacteria or form of life that was on there to grow into its own colonies. So you can observe them on the plates. Each one of those little dots is actually a colony of bacteria. So just from looking at the pictures, you can already see a striking difference. So the top row is all of the top halves of the pupal cases, and there's not a lot of growth. And on the bottom halves, they're so dense that it was actually, we had to count them under a microscope. And there was also a lot more color, so we had more red and yellow colonies popping up.
And so from like a visual inspection, there's definitely something going on. There's definitely a difference between the pupal case that has some meconium on it and the part of the pupal case that doesn't. So we counted all these colonies and graphed the data sort of potentially support just a visual inspection we did. So this bar graph shows the bottom of the pupal case on the right and the top on the left. And there's definitely a greater abundance of colonies on the right, which is sort of visual from the photos. The plates on the bottom are definitely a lot more dense than the plates on the top. And then we looked at sort of the differences in the types of colonies or morphotypes. And even just from the visual inspection, there's all these red dots, which weren't really, they weren't really part of the top plates. So our data supported that, having red, sort of white and yellow colonies that weren't as present on the top of the plates. And so sort of condensing our findings based on these experiments we did, we found that there is sort of research that we did to suggest that there is microbial activity around the meconium, and that further research would definitely be important to see if there's any function of this bacteria around the meconium, because we were sort of researching to begin with whether or not there were issues having these, the feces with the pupa in the colony, and whether or not that could be an issue with disease.
So when applying this to human systems, we were actually kind of surprised by our results. So when you think of like a human hospital, when a baby is born, everything around it has to be extremely sterile and bacteria-free because they're so vulnerable at that age. But what we saw with the ants was that they were actually encouraging microbial growth on their most vulnerable population in the colony. And so we've seen recently a lot of research coming out about antibacterial products not being effective and also potentially causing problems where bacteria become resistant to our products, and that makes them too resistant for us to create solutions for. So we think that this is an interesting way of embracing bacteria and not trying to eliminate everything.
So to sort of conclude our presentation, I think we can sort of take a lesson from ants and the natural world and how they potentially use bacteria to their benefit for different functions within their colony, and to their advantage, and potentially apply that to a human system, whether that's sort of washes that are probiotic, or just thinking about textiles and products on a human scale that don't necessarily sort of get rid of bacteria, but potentially promote good bacterial growth. Thank you.
Thank you.
Awesome. Great job, guys. So yeah, so that's what we're dealing with. I'm really impressed by the students and their sort of embrace of pushing the scientific boundaries forward and then thinking about how that applies to their work as a non-scientist. And I think that's the power of biologically inspired design, is that it's this idea that people outside of science can even embrace science and sort of look at it as an exploratory sort of innovative ideas for bringing to their own work. And I think that's a really sort of underutilized a portion of natural history research, and one that I'm proud to be a part of and proud to have at the museum here. So with that, we can take any questions. If you have questions for the students or questions for Clint and I as instructors, Clint can come up here with me and answer some questions. Come on, Clint. You got to get up here.
So yeah, if you've got questions, raise your hands. That way I know who to bring a microphone to. We'll bring a mic around. That way everybody in the room can hear the questions.
So I have a sort of a general question. So I come from an art background, and you're merging art with science here. When I was looking at the image of the circular part in the campus, and you were talking, what was the program? It was Stigmatize.
Yeah.
So when I looked at that, I thought about a petri dish. And then that you took the action of actually trying to provoke other people to follow in that version, and it made me think about putting your thumb in the agar in a petri dish. So what I'm curious about is whether you find tension or complication between the exactitude of science and following that kind of methodology and design, which is just pretty much freeborn, perhaps anarchistic in its approaches. What do you think about that?
Well, so one of the surprising things about doing a course like this, and I think something that the students all experience, is in this context, science was the broad thinking. Science was what's outside of the box thinking for their process. Normally, it wouldn't be in their process, but we sort of threw it in there and said, can you make this applicable? Can you do real science to sort of push the knowledge forward, and then can you take those ideas and use them as sort of spurs to go out and do innovative thinking? So I think it was kind of cool because it's sort of the reverse of that. It's science as broad thinking and putting it into their known and honed sort of expertise as designers. I think was really, really cool. Another part of that project that they didn't show was they actually had a failed experiment in that, where they put out just folding chairs, and people did nothing with those. Yeah, which is kind of interesting. It's kind of interesting to think about, but the only stigmatizing thing that had an effect was using their physical bodies as the trace in the environment, about how to use the environment. Even when you put out chairs, people just didn't sit in them. They had to be out there, which is, I think, really cool. Yeah. So it was more about using the scientific method to sort of think about ways of proofing design and bring that sort of stuff into their design.
So I have a question for the students, especially the senior, the ones that are getting ready to go out in the job market. Did this class influence or change your career direction and maybe think about incorporating or partnering with science if you're pursuing like a job in the design field?
I think in the world, especially in architecture, because that's what I know the most about, but you do work with scientists, you work with structural engineers, you work with different consultants. If you're working on maybe a hospital, people who know about antimicrobial properties of spaces and stuff. So it's very connected. And I think there is sort of sometimes a misconception about design and that it's purely artistic or purely for visual purposes, but it's for function. It's form follows function. That's like a common philosophy in design. You're taking these principles that you learn from science, you learn from other fields and clients, and you're using that information to build something functional and purposeful. So I think it kind of goes to show in design just how broad it is and how broad of a field it can be. So yeah, I think it's just inspiring to keep moving that direction.
I'm in industrial design. My name is Dipali. And yeah, I think definitely making a point to your question, I think it helps designers as well. I think designers get a rep of being kind of over generalists, which we can be. And it helps us kind of hone in on these really specific skill sets and really tangible experiments that we can work with and come up with an overarching principle. And I'm very into scientific and medical design. And I think science is an incredible vehicle for studying behavioral design as well, like some of our group mates did with the Stigmergy project. So yeah, it was a good experience overall.
So this was really, really cool stuff. And I guess my first small question is, are you going to do it again? The course.
Yeah, I believe so. This is our first offering, so like I said, it was experimental. So normally courses aren't taught like this. Normally it's not based in bringing design students into the lab and having them explore unknowns. It's more working with knowns. I think Clint has taught several classes about this at Arizona State, actually. And so I think for both of us, I don't want to speak for you, but
yeah, so I work at Arizona State University teaching similar classes within the design school. And there, it's always been 100% designers, usually at the graduate level, who start with a design problem. So they might start with like rain gathering in a building, and then they'd go look at leaf shape and stuff like that, or filtering systems like whale, baleen, and see if they can develop something from that. But what we ran into after we taught these courses a couple of years is that the students kept hitting on the same examples because that's basically what's out there. They're limited by the research that's out there and how it's been presented to them. So one of our goals with this class was to get designers' hands a little dirty. They did microbiology, which was kind of crazy, and get them engaged with science and to where they could actually read scientific papers and start to see where interesting things might turn up if you push a little bit deeper. So I think with this class, it was just interesting to see how all the projects came up with things that were completely new, whereas they weren't turning to these pre-baked biomimicry ideas that so much of the field has relied on in the past.
Well, so one follow-up. If I may have misheard, but in your opening remarks, you were talking about design being inspired sort of by not only by the natural world, but the scientific process. But I thought you also said that the design process might influence the science, which I may be interpreting not quite right.
Right.
But I'd be curious to see that side of the
yeah, so for us, we work together a lot just doing ant science projects. And this, like literally these projects push the boundary forward on things that we were actively working on. So some of the nest architecture that we got out of this is now become a research project in my lab. We found some really cool, like the things that Hannah and Kevin ended on, showing the new nest architectures that were nothing like what we thought they should be. That was like mind-blowing when we did those for us, from Clint and I. And then some of the Stigmergy stuff, the students actually visualized in those videos walls appearing. They were excavated and then they were refilled, and you could watch that in real time. That no one's seen that before. that hasn't been seen. So that's just something that goes forward in our research and legitimately was new scientific progress that they made, which I think was just awesome. Yeah.
Another thing to add that too, the cuticle structure has been a low simmering project for us. We've been studying how ants deal with disease, looking at how the antibiotic compounds ants produce and stuff like that. But we started to realize there might be something going on with these weird patterns on the cuticle, and literally they have never been studied by a biologist ever in ants. We don't know what they do. And so we thought, well, okay, maybe they actually do things like prevent bacterial growth. And this was great. The students actually pretty quickly showed us that no, they absolutely do not. There's no way that's happening. And that shifted. Now we think that these ridges might have something to do with culturing beneficial microbes. So now we're culturing microbes from different ant species as we speak. We have some stuff going on in the lab. So in some ways it helped us do some R&D as we continue our research projects in the future.
I'm interested in your ant colony models that you put there and comparing that with cities of the world. Of course, humans get to talk to one another and see how one city is designed. But I'm interested, different species, I assume, of ants have different types of colony tunnels. But if we have the same species and they're in another continent, how in the world can they tell one another how to design those tunnels?
So I mean, it's an evolved response, and so they don't tell. For ants, it seems that the architecture types are species specific. We totally believed that until recently. We did 2 sites of the same species and found out there's some differences. But it seems that they converge on similar plans within a species. But what's been interesting is that when you start to classify them, there's patterns you see across ant evolution. And so some of them do things like they build a shaft that's straight down, but if they want to get bigger, they have to add a shaft. And there's weird things like they're not connected at the bottom. But if you look at a fire ant colony around here, they're a mess. They're connected all the way through. And so we're thinking that has something to do with how they control traffic flow through the nest, similar to how we're thinking about traffic flow through cities. Another one, again, I've talked about disease a lot, but that's another thing we're really interested in. How disease spreads in a city that's designed like a spoken wheel versus one that's designed like a linear system might have some parallels to how a pathogen spreads in an ant colony if it's designed like a fire ant where all of the individuals are interacting, or if it's designed like one of these harvester ant nests where if you live in one tunnel, you don't necessarily go to the rest of the nest. There might be more confined systems with disease dynamics and stuff like that.
Hi. Well, first, thank you for presenting on this. I have a question about climate change. I'm just wondering broadly, how do you think ants and certain ant species are going to respond to harsh and rapid climate change conditions?
He's a good person to ask about that.
Yeah, so I actually, most of my research right now, a lot of that is based on climate change and how temperature affects ants. And it depends, right? So some species do well with warming, and so they're going to do better. Things like fire ants here, right? Fire ants are pretty thermal loving. So as it gets warmer, they might be doing better than some of the more native species that deal better with cooler temperatures. But it could affect things like nest architecture, so they're adaptable. So when you look at something like a nest, they move throughout the day to track preferred temperatures. And the way the chambers are designed or where they're located in the soil layer probably depends on temperature to some degree. So there's plastic responses. They can affect how they build, so they move to cooler areas of the soil rather than warm areas. So in some ways, they're buffered from temperature change, especially from the negative impacts of heat compared to some solitary species that aren't buffered by the social structure and the architecture they build and stuff like that.
So how do you think we may learn from them?
That's a good question. We didn't address that at all in this. I mean, one thing that ants can do is move, and so climate change is really a human problem, right? We're the ones who built giant cities out of concrete and marble on the coast where we're going to have sea level rise, and it's going to be a real problem. If you're an ant and you live on the coast, maybe some colonies will die in a storm surge, but the populations over time are just going to shift further back. So maybe looking at some of that adaptable behavior that they have and trying to incorporate it into what we do could be beneficial.
So I have a question, and it's something that actually someone who works in your lab, Adrian Magdalena, talked about in a talk she gave not too long ago, but that there are species of ants that form super colonies that stretch huge areas of land. And so I'm wondering what the inspiration might be in relation to human systems and human activities for comparing us, or comparing ants to us, or the other way around, for these vast areas covered in ants.
Yeah, that's funny. You bring up super colonies. In my mind, super colonies are pretty menacing because they usually end up being invasive species. That's why they're super colonies, a lot of them. Because
That's a great metaphor.
Yeah, yeah. So I don't have a good answer off the top of my head, but I do know that super colonies, they usually lose that ability to distinguish colony boundaries. In some of the cases, they've lost that ability to distinguish chemically which ants belong to which colony, so they don't fight anymore. So there's free movement between colonies. So it has a lot to do with their communication systems and how they achieve the level of super colonies. But in some cases, it's also from a genetic bottleneck. They'll arrive in some new place because of us, because we ship them there in some sort of cargo ship or a container. And then there's genetic bottlenecks, so there's not that genetic variability, and they lose that differentiating ability. I don't know. Do you have a better answer?
I mean, one thing that I would add to that, I think it brings up a good point, is that when we talk about biomimicry, we don't necessarily want to copy everything. We don't want to build cities that look like ant nests. I mean, ants, when you get down to it, they can be pretty nasty. They have warfare. I mean, there's ants that enslave other species of ants, and there's all these terrible things. So when you look at a field like biomimicry, what we're doing is trying to look at things in nature that can inspire us in ways that are more sustainable, or that could benefit us, especially by abstracting them and using them in a way that applies to humans. But something like a super colony might be, we don't want to. I mean, maybe some humans want to develop a horde that conquers the earth.
Why'd you look at me when you said that?
Yeah. I'm not going to wade into the politics here, but I think it's good to point out that we don't want to. If our societies looked exactly like ants, we would probably be worse off than we are today.
Let's give everybody one more round of applause, especially for our students. It's a brave thing to get up in front of strangers and talk about what you've worked on for a whole semester, sort of off the cuff and the way they had to do it. So applause to you guys. You did a fantastic job. Great work. And thank you all of you for coming out to the Science Cafe tonight. If you loved it, do me a favor. There's a feedback form on every table and some pencils for you to fill it out. It's actually a brand new feedback form. We revamped it. There's new questions on there we want you to take a look at. Give us your feedback. Leave your contact information there so that we can let you know about the next round of Science Cafes. When they hit the schedule, you'll be one of the first to know. That way you can go ahead and mark your calendars for every Thursday night right here at the Daily Planet Cafe. Be a great help to me and to the museum so that we know that you love it. And if you didn't love it, just say that you did. Everybody have a great evening, travel safe, and we'll see you next week here at the Science Cafe. Good night, everybody.